Good evening and welcome to the Gloucester Marine Genomics Institute's November Science Hour. I'm Chris Bolzan, GMGI's Executive Director. Tonight, we are excited to hear from two top coral scientists who are joining us from Mote Marine Lab and Aquarium virtually from Florida. In reviewing our registrant list for tonight, I saw dozens of names new to our organization. Welcome. We are so glad you have tuned in. And to our loyal Science Hour followers, we're grateful that you continue to join us each month and share our passion for science education. For those of you who are new to GMGI, we address critical challenges facing our oceans, human health, and the environment through innovative scientific research and education. By bringing world-class science and transformative workforce development to Gloucester's historic waterfront, GMGI is catalyzing the regional economy. You can learn a whole lot more about our research projects. You can meet our team, read our news, get a sneak peek inside our brand new biomanufacturing learning environment at our Gloucester Biotech Academy, and you can watch prior science hours and so much more if you take some time and explore our website, gmgi.org. Tonight, I encourage you to please use the Q&A function at the bottom of your screen for any questions that you might have for Doctors Muller and Koch. And thank you all for tuning in and for continuing to share our excitement for our oceans and science education. Thank you also to our 2021 Science Hour presenting sponsors, Ms. Elizabeth Moore, the 1911 Trust, and the James and Gail Bacon Family Trust. I'm going to turn the screen now over to Dr. Andrea Bodner, GMGI's Donald G. Combs Science Director, who will introduce our talk. Andrea?
Thank you, Chris, and good evening, everyone. Thanks so much for joining us this evening. I'm Andrea Bodner, the Donald G. Combs Science Director at GMGI, and it's my pleasure to introduce tonight our two speakers for tonight's Science Hour, Dr. Erinn Muller and Dr. Hanna Koch. Erinn is the Associate Vice President for Research, Senior Scientist and Program Manager of the Elizabeth Moore International Center for Coral Reef Research and Restoration at the Mote Marine Laboratory in Sarasota, Florida. And Hanna is a postdoctoral research fellow at Mote's Elizabeth Moore International Center for Coral Reef Research and Restoration. Erinn completed her undergraduate studies and master's degree in marine biology at the Florida Institute of Technology, and earned a PhD in biology, also from Florida Institute of Technology. Her research is focused on coral health and disease, where she uses a variety of techniques to understand the causes and consequences of coral disease outbreaks, as well as the mechanisms that promote coral health and resilience to major stressors such as temperature and ocean acidification. Her work has shown that stressed corals are more susceptible to disease, and she anticipates that once we understand the mechanisms leading to compromised coral health, we can prevent disease outbreaks in future. Hanna conducted her undergraduate studies in marine science at Eckerd College, her master's degree in biology from California State University at Los Angeles, and her PhD in evolutionary biology from the Max Planck Institute for Evolutionary Biology in Germany. Hanna joined the Mote Marine Lab in 2018 as a visiting research scientist with the German Research Foundation to integrate assisted coral sexual reproduction into Mote's ongoing coral reef research and restoration framework. In 2020, Hanna became a postdoctoral research fellow at Mote to continue her research on coral sexual propagation, assisted evolution, and coral reef restoration in the Florida Keys. Tonight, Erinn and Hanna will be sharing an update on their critically important work to restore and preserve Florida's imperiled coral reefs. So with that, I'll turn things over to Erinn.
Thank you so much for that wonderful introduction. It is a real honor and pleasure to be able to share with you today kind of a 10,000-foot overview of our coral reef research and restoration initiatives at Mote Marine Laboratory. And the work I'm sharing with you today is a reflection of what I do, but also a reflection of what our 30+ staff at Mote that focus on corals have been doing for the past decade or more. So although I am stationed in our main campus in Sarasota, Florida at Mote Marine Laboratory, we have an amazing, there we go, an amazing infrastructure that's a sister campus off of us down in Summerland Key, Florida, close to Key West. And this infrastructure is 19,000 square foot of a Category 5 hurricane resistant building that houses dry labs, wet labs, classrooms, and space for visiting scientists to stay, as well as our 25 plus staff that focus on coral reef research and restoration. And then surrounding the outside of the building, we actually have one of the world's largest land-based coral restoration nurseries. And then several miles offshore of our infrastructure, we actually have 2 underwater nurseries I'll show you some nice images of that grow tens of thousands of corals. And so why, as an institution, do we focus so much of our people power, our resources, our funding towards coral reefs? And it's because coral reefs are so incredibly important for our oceans. They're estimated to be the most biologically diverse ecosystem, where potentially up to 25% of marine life rely on reefs in one way or another during any stage in their life cycle. They also provide a suite of ecosystem services that are really important for humans, whether you recognize them or not. They're happening every day. And one of those ecosystem services that these reefs provide is shoreline protection, because as waves roll through, they break on top of those coral reefs that are offshore of our land, of our roads, of our infrastructure, and they actually prevents erosion and protects all of that infrastructure that we've placed on land near shore. They also provide a novel source of compounds to fight diseases. So today we harvest a lot of compounds from organisms that live only within the oceans, and we use them to fight human diseases like cancer, drug-resistant bacteria, and also Alzheimer's disease. And also, at least 1 billion people around the world rely on reefs for food or income, and so they're a source of economic and societal well-being. Taking it closer to home, you can also see the incredible value of Florida's coral reef, which spans from Martin County and the central east coast of Florida all the way down to the dry Tortugas. And that structure in itself is a huge economic foundation and driver for the state of Florida. It helps to attract over 16 million visitors every year, supports at least 70,000 local jobs, and it's estimated to be worth at least $8 billion to the economy. And so it's a huge asset for our state and for the entire continental United States. Unfortunately, we're losing coral reefs at an unprecedented rate. If you dove Carey's Fort Reef in the Florida Keys in the 1980s, it was an icon of what healthy coral reefs should look like. It was dominated by 2 species, the staghorn coral that you see here in the front and the elkhorn coral in the back. This reef was covered at least in 50% with those stony corals, which is kind of the characteristic that we use to quantify a healthy reef ecosystem. Unfortunately, now if you revisit the same reef, it's a completely different phase shift. It's the skeletons of corals past that you can go and see. Basically, it's the skeletons of corals that used to be alive, and that's all that remains, where there's plenty of sediments and algae and some gorgonians or sea fans, but not the living stony corals. And so now this kind of phase shift that we've seen here in an example with Carey's Fort is happening all over the Florida coral reef, but also all over the world. And unfortunately now Florida is an iconic emblem of how we are losing corals and coral reefs at an unprecedented rate. However, Mote, as an organization, recognized over a decade ago that in order for these reefs to start coming back, we had to actively engage and intervene and assist these corals in their ability to come back to a self-sustaining population. Although there were protections in place through the Florida Keys National Marine Sanctuary, there was no evidence that these corals were coming back on their own. And so about a decade ago, Mote established our first in-water staghorn coral nursery. And what you see here are these underwater trees that are created by PVC ropes and floats. And instead of kind of pine needles dripping off the branches, you have individual coral colonies. And we have 2 of these in-water nurseries now that are mainly focused on staghorn corals, but actually house a suite of different species now. And we can have over 20,000 of these corals growing within these locations at any given point in time. Now, what we do is initially we started using these staghorn corals because they were easy to grow and they were easy to outplant. And how we outplant them is really quite simple. We take little fragments from those underwater trees and we take them in, place them out onto the reef by taking a masonry nail, hammering it into the reef structure, and then attaching the coral adjacent to that nail sticking up. And that coral will actually grow over the nail and reattach to the substrate within probably about a month. And as you can see in the image on the left here, we outplant them kind of in close clusters of 5. And all of those individuals actually came from the same parent colony, meaning they have the same DNA. This is really important because as you see in the image on the right, which is about a year later, those corals have grown to be what we call a little thicket of staghorn corals. When they touch each other, if they are the same DNA, they'll actually fuse and will create a large coral within just a year. And this is important because corals reproduce based on size, not age. So we have to hit a threshold in size in order for them to be reproductively viable. And Hanna is going to be talking quite a bit about that in the next talk. We've gotten really good at figuring out how to outplant them and how to get them to survive in the short term. On average, we have over 95% survival of our staghorn outplants within that first year. So they're able to withstand the transportation stress, the initial acclimation that they have to do to the site, and grow and fuse. And so we've been able to be really successful with this as well. But we knew that we couldn't really recover reefs as a community by focusing on one species. So we kind of, cut our teeth on that one species, figuring out how to best grow it and outplant it, but then realized, all right, now we've got to shift our focus to some other species that are critically important for all those ecosystem functions that reefs provide. And these have been deemed kind of the orphan corals of restoration, especially the boulder and the brain corals, because they grow so slowly, only a couple millimeters a year. And so it was very difficult for restoration practitioners to get a grant or get funding and have significant progress to show for that effort within just a couple of years because of the slow growing nature of these corals. However, we developed a process that we call microfragmentation and reskinning. And so Mote scientists, over some trial and error, actually figured out that if you take a large piece of a coral and use something like a bandsaw and cut it up into tiny, about 1 centimeter squared pieces, that process of fragmentation accelerates the growth. And the corals actually move a lot of energy into growing that tissue back that it's lost through the fragmentation process. So we can actually get those corals to grow up to 50 times faster than they normally would if kind of left to their own devices. Now we cut these corals in a different way, creating those microfragments, and we actually attach them to a ceramic plug or a disc like you see here. Now to outplant them, once they reach the entire size of that disc, about 3 centimeters in diameter, we move them out onto the reef. And there we can take the discs, and instead of kind of having branches in a cluster, we actually outplant them in clusters of discs, anywhere between 5 and 15 clusters. Now, when we outplant them, they can kind of individually, we outplant them in a cluster of the same individual, again, with the same DNA, so that a year later, they actually grow over that reef substrate, touch each other, fuse, and then start growing up and creating branches, all within about 12 months. And this is an example of an elkhorn coral outplant that showed some really nice growth and survival after that outplanting process. And as I mentioned, we have one of the world's largest land-based coral nurseries. Within our nursery on land, we have about 100 of these blue rectangular raceways that hold about 1,000, up to 1,000 corals each. So we have about 30,000 fragments total that we usually are growing and propagating within our land-based nursery. They represent thousands of different genotypes. So we have a lot of genetic diversity that we integrate into our restoration plan. And we outplant about 10,000 of these corals a year. And so I think I skipped over the staghorn stats, but we outplant about 20,000 of those staghorn corals a year. So right now we're outplanting about 30,000 corals back onto degraded reefs every single year, growing basically exponentially in our production each year. And I mentioned we had good short-term survival, but we also have some really nice long-term, like ideal success stories that have occurred because of our restoration efforts. So one of our success stories is Eastern Dry Rocks. So this reef had absolutely zero staghorn coral on it when we started outplanting there. After 3 years, we still have over 90% survival of the corals that we put out, and that we created over 700% more living coral cover just by doing the initial outplanting and allowing those corals to grow and recover over 3 years' time. We also try to quantify the other ecosystem benefits that may be occurring because of the pure practice of putting corals out there. And so we're looking at herbivory rates and fish biomass to look at how outplanting can influence other organisms that are interacting with the ecosystem. And also how our outplanting efforts can influence the rugosity and the net accretion of those reefs over time. We know that shoreline protection is critically important. And as reefs are degrading, they're becoming flatter and flatter, and they're sinking actually as they become more eroded. And so we need to ensure that those reefs keep pace with their natural capabilities and also with any sea level rise in order for that ecosystem service to be provided for shoreline protection. And so we are quantifying how our outplanting efforts can influence those accretion and shoreline protection efforts, and also how we can influence self-recruitment. So this is a little image of a settlement plate. We want to get the populations back up to a place where they're self-sustaining, and we basically put all of our scientists out of a job. And so making sure that they're reproducing and settling back into that reef environment is really important goal for us. And then finally, we also integrate a lot of science associated with resistance to major stressors affecting corals today. So we want them to survive today, tomorrow, and next year, but we also want them to survive for decades to come. Here's an example of how corals can respond differently to stress. You actually have two genotypes here. So two corals with different DNA, one on the left, one on the right, and they look very similar in this picture. But a month later, after experiencing just 2 degrees Celsius warmer water temperatures, you can see a very different physiological response of these corals, where the genotype on the right is highly susceptible and is doing what we call bleaching, spitting out its algal symbionts that are critical for the coral to survive and live, whereas the coral on the left has maintained that symbiosis and can actually recover and survive long term. And so we're trying to integrate the knowledge that we can pick and choose which corals we're upscaling within our restoration based on their resilience to some of the traits that we know unfortunately aren't going away, such as increasing water temperatures, disease, and ocean acidification. So we actually bring all the different genotypes, or at least a subset depending on how many we have, into the lab where we can manipulate their exposures to things like high water temperatures, pH through ocean acidification processes, and even disease pathogens to figure out which ones are going to survive these major threats and which ones are not. And you can see the physiological responses associated with that. So this is the 2 different genotypes under the same environmental stressor, high water temperatures, where the one on the right has grown significantly. It's much bigger than the one on the left, and it actually has retained its coloration. That means it is more resistant to things like climate change. And we want to utilize that information into our restoration practices in a couple different ways. One, we can potentially upscale the production of certain genets so that they're more represented at the sites that we're outplanting in. But we also are very cognizant of genetic diversity and how important that is for population level resistance and adaptation, which Hanna will go into in detail. And so we can use them to create more resilient offspring. I won't go into this very much because Dr. Hanna is a specialist in this area and has a lot of really cool stuff to share with you on what we're doing here. But a lot of the information that we conduct on the resilience side of the screening funnels directly into Dr. Hanna Koch's research on coral reproduction to help create more resilient restoration corals. And I just want to touch on a couple different initiatives we're also doing at Mote, which I think are incredibly innovative and timely. One is our Mote Marine Laboratory's International Coral Gene Bank. This is a hardened infrastructure 20 miles inshore of the water from Sarasota, Florida. It's basically a warehouse of these raceways that we have created to be a Noah's Ark of, right now, Florida's coral reef. So we are focusing on obtaining all the species within Florida's area and as much genetic diversity as we can possibly contain and have them within the warehouse in duplicate to maintain corals for repopulation of the reef so that they're in a protected safe haven while nature continues to hopefully get healthier for these corals to be able to persist out in the wild. But what's really cool about this infrastructure is not that it's just a warehouse and a holding facility, but it actually has a spawning room adjacent to it. And so many of our corals are large enough to be reproductive. And so we filter these corals into the spawning system, which allows them to queue into things like the metagenesis and allows them to spawn. And we can create babies from these corals that are in our ark. And those babies are going to funnel into our coral restoration program down in Summerland Key. So we can utilize this infrastructure to be a Noah's Ark, but it also can be a catalyst to create genetic diversity and species diversity that we can use for restoration. And also, there's a huge effort led by NOAA, National Oceanic and Atmospheric Administration, called Mission Iconic Reefs. It's an effort to do a comprehensive restoration initiative in 7 iconic locations throughout the Florida Keys to bring these reefs back to 25% of their coral cover that they previously had in the 1970s. And Mote is a major partner associated with this effort. And so over the next 20 years, we'll be working quite heavily on these 7 reefs to make that mission a reality. And finally, I'll just close my section by saying that one thing I'm incredibly proud of related to Mote and our restoration program is that we really think about all the different steps and facets essential for conducting effective and resilient coral reef restoration. And we can do them all within one organization. And it's incredibly great to have partners and collaborators outside. And we have hundreds of them. But it's also really nice and really efficient and effective to be able to conduct this entire cycle of sexual reproduction, asexual fragmentation, resilience testing, propagation and nursery grow out, outplanting, and then completing the cycle by using those corals to be the next generation, all within the same roof. And it increases efficiency and allows us to be really effective in our methods and our missions. And with that, I would just like to thank some of our major sponsors of effort from the federal agencies, our state agencies of Florida, and also a plethora of foundation and philanthropic support. And now I will hand it over to you, Hanna.
Hello. Thank you, Erinn, for that introduction. And thank you, GMGI, for having us. I will go ahead and share. So for my portion of the presentation, I will talk about how I use assisted sexual reproduction to restore Florida's coral reef. And let's see if I can get rid of, one second. There's a bar at the top, so I'm hoping you can all see my slides.
It looks good from our end, Hanna.
Okay, good, thanks, because it's obscuring some of my slides for me. So as long as you can see it, that's good. So reef building corals, also known as stony corals, reproduce both sexually and asexually. In nature, asexual reproduction via fragmentation serves as a means for population growth and expansion, where a portion of a colony will break off and then reattach to the reef and form an entirely new colony. But importantly, that colony is still genetically identical to the parent colony that it came from. So we call it a clone. And this is the basic idea behind the coral gardening tenet of branching coral species, where we can use the fragmentation process to create thousands of new colonies quite quickly and rapidly for using for active restoration. So on the left, you can see our coral nursery where those fragments are produced and grown out, and then we'll affix them to a degraded reef. And then over time, as Erinn already discussed, they'll grow and form these large thickets. And in this way, we can quickly increase live coral cover, as well as available habitat and shelter for other reef organisms like fish. And as Erinn also described previously, Mote scientists previously further developed this methodology for being able to apply it to the species that don't tend to naturally fragment as easily. So these are the slow growing massive species, the boulder corals, the brain corals. And instead of breaking them, they'll use a saw to cut them into these replicate microfragments, then grow them out and then outplant them onto these degraded coral heads. And in this way, we can actually restore centuries old coral heads in a matter of a few years. And the other outcome of this is that we can actually create larger colonies faster. And this is actually really important for the sexual development of these species, because as Erinn mentioned also, sexual maturity is size dependent. So this means that the colonies have to reach a certain size before they can produce that next generation. And that's really critical to the survival, the persistence, and the adaptive potential of these populations, because it can take years or decades for these colonies to reach this critical milestone in their life and in their life cycle. So anything that slows it down or prevents it from growing to that stage actually prevents that reproductive cycle. And so what Mote has shown is that through this microfragmentation fusion methodology, we can actually cut this process down to a fraction of the time. So instead of decades, in just a few years. So with these images, they show actually the first of Mote's mountainous star corals, which is a very slow growing species, to reach sexual maturity last year. So these outplants were about 5 years old, and in the weeks preceding their annual predicted spawning window for the species, I went out and I took some core samples, and I was actually able to positively ID developing eggs and sperm inside of these colonies, which indicates that these corals were ready to become parents for the very first time. Then a few weeks later, I was able to document them spawning. So I'll show a short video, and it's a very quick video, but what you'll see is the instantaneous release, a process known as broadcast spawning, of these gamete bundles, which are actually the packages of the eggs and sperm, because this species is a hermaphrodite, meaning it's both male and female. So even though this is a very short event, it's actually a very significant event and actually represents a science breakthrough for the coral reef restoration field. And that's because these are the first corals of any slow growing, massive or mounding species documented to sexually reproduce after being restored to the reef. This is a significant beacon of hope for coral restoration worldwide, because this methodology can be applied to any species or region of the world. And it also showcases that Mote's resilience-based restoration strategy works. These corals within those 5 years survived multiple bleaching events, as well as a Category 4 hurricane, and one of the worst disease outbreaks that Florida's coral reef has seen. So most stony coral species actually sexually reproduce through the broadcast spawning process, where they'll release their gametes, their eggs and sperm, into the water column during these annual mass synchronized spawning events, where the sperm and eggs will fertilize each other to produce larvae that eventually settle back down onto the reef through a process called recruitment. And after they recruit onto the reef, they undergo metamorphosis, where a primary polyp will be formed. It'll grow, divide, form new polyps, essentially creating the colony. Now, importantly, in this process, every new individual, every new sexual recruit is genetically unique. So there are many benefits of sexual reproduction to the survival, the persistence, the adaptive potential of populations. And one of them being that it replenishes depleted adult populations and promotes population recovery after disturbance, like a bleaching event or a disease outbreak. Coral larvae can also disperse to new areas and reefs on ocean currents, which support really important processes related to gene flow and population connectivity. And just as in humans, sexual reproduction in corals shuffles genes, resulting in offspring with different combinations of traits inherited from parents. So this random shuffling process is a really important source of genetic variation, which in turn serves as a mechanism for populations to cope with environmental change or stress. So imagine a genetically identical coral population where all the colonies are clones of one another, produced maybe through this fragmentation process. And imagine how that population will respond to a novel disease outbreak compared to a genetically diverse one, where within the clonal population, if that genetic variety is susceptible to that disease, that population is more likely to go extinct. Where in a genetically diverse one, you're more likely to have individuals with different traits or combinations of traits that will allow them to survive, persist, and carry on the population. But unfortunately, what we're seeing for many of our most important species on Florida's coral reef is that new sexual recruits are failing to show up. We're not seeing the next generations of these corals showing up on our reefs. And so there could be many reasons and many points in the sexual cycle where these failures are happening. First of all, if corals are spawning in temporal isolation. So for corals, they rely on a very specific set of environmental cues to regulate the timing of spawning throughout the year and even within the month, and also related to like the full moon. And so any disturbance or stressor that disrupts these triggers can actually throw off this timing and cause the corals to spawn at different times. And if corals spawn at different times, fertilization will likely fail because gametes are only viable for a few hours after they're released. So if the corals are spawning on different nights or even a few hours apart, fertilization will not be as successful. Also, if the corals are spawning in physical or spatial isolation. So unfortunately, many of our populations are so degraded that the corals are really spread out and sparse, and they're not dense enough to effectively support successful fertilization because it's just too far for these tiny microscopic gametes to find each other in the wide open ocean. And then also what we've seen in our region is a shift from coral-dominated reefs to algal-dominated reefs. And essentially what we have is a shortage of available niche for these very delicate, sensitive coral larvae to settle and effectively undergo metamorphosis, develop, and survive. So in order for the benefits of sexual reproduction to be realized for our populations, we basically have to step in and bring this process into the lab and carry it out by hand. And so in this way, combining assisted sexual reproduction with resilience-based restoration strategies, genetic management, and effective outplanting methods, we can actually accelerate population and reef recovery, which is our overall goal here. Before we can actually produce the babies, though, we have to get our hands on sexually mature, reliably spawning corals, which might sound like a given, but unfortunately in our area has posed a major challenge. And so I've devised a number of different strategies to try to address this. One way is I've created in situ spawning nurseries where I allow the corals to grow in optimal conditions and unfragmented so that they can become sexually mature. And then we'll bring them on land for annual reproductive events. We also have located a few reefs that have a few remaining wild colonies that are sexually mature, and we can conduct in situ spawning and gamete collection. But this is very risky because spawning season in this area coincides with hurricane season. And so, these events only happen once a year. And you're putting all your eggs in one basket and relying on this event to happen, but a hurricane can come through or bad weather conditions can basically preclude these operations from happening, which has happened many times. So to combat that, we actually have installed a state-of-the-art ex situ spawning system, which allows us to replicate all of those environmental conditions that corals need to develop and release those gametes. So we can have greater control and manipulation and reliability with using these systems. And then of course we can create our own sexually mature corals using this microfragmentation fusion method in the lab. And we also partner with a number of amazing other institutions and organizations to exchange larvae every year in order to maximize our efforts and diversify even more. Once we're able to actually get the corals to spawn, one of the major themes of my research is understanding broodstock compatibility. So two main themes within that are the different genetic varieties spawning synchronously? Are they spawning within the expected time frame after the full moon? Are they spawning during the expected time frame after the sunset? Are the replicate colonies of the different genetic varieties spawning at the same time? Are the different genetic varieties spawning during the same time each year? And then we can assess reproductive compatibility by looking, by actually conducting different two parent controlled crosses and assessing fertilization rates. And so if we see certain crosses or certain combinations of parents consistently yield low fertilization rates or high larval mortalities, we can swap those genetic varieties out to make sure that our broodstock pool is always optimal. And then I'll also conduct managed breeding, and this is where I will incorporate Erinn's work on resiliency and stress testing, where after she's already screened the different genetic varieties to see which may be more disease resistant or stress tolerant, I'll use them as parents in specific breeding designs to create genetically diverse, stress tolerant offspring with different traits that we can use for restoration and use for research as well to understand the heritability and the transference of those traits across generations. So after spawning, there comes settlement, and there are a multitude of factors that play into the successful settlement of coral larvae. And unfortunately, they're very species specific. So not one size fits all is a strategy here. And a lot of these factors you have to test for the individual species. So every year that I do this spawning work with the different species that I work with, I'll set up a series of studies looking at different factors like optimal setup design, optimal larval stocking density, which are the better chemical or spectral cues that can attract the larvae to settle where you want them on the different substrates, the different types of substrates, the levels of conditioning and orientation as well. Ultimately, what we want to do is make sure we're achieving as high settlement rates as we can. And then after settlement, we have these microscopic, tiny, little, very delicate, sensitive babies that we have to keep alive for a very long time. So we have to make sure we're providing a very healthy environment to optimize growth. That way we can use them for research and restoration as quickly as possible. So we'll optimize all of these different factors and make sure that we're giving them the best possible conditions so that they can survive and grow and be used for further research and restoration. So presently, we're in the process of sexually propagating 6 key stony coral species. And once these coral babies reach a certain stage, we can actually use them for the various restoration and research projects, of which I'll go over a few. So regarding restoration, once the corals will become a certain size, so when they cover the surface of the plug or the substrate that they're on, we'll insert them into parallel propagation pipelines, both in situ and ex situ, basically where they're repeatedly fragmented and outplanted. And we also, during outplanting, we make sure that we set up our populations, our restored populations, for reproductive success in the future. And so we do this by outplanting genetically diverse, dense outplants that should reproduce sooner through the mechanism that Erinn already described, where we will use replicate fragments and outplant them near each other, and their clones, they'll fuse and form a larger colony faster. So in this picture here, this is an example of a site restored through assisted sexual reproduction. Each one of those little plots of 5 corals, each one of those plots is a different genetic variety. And within the plot, each of those pieces is a replicate fragment. So these images show our first example of the staghorn sexual recruits that we outplanted in February of 2020. So on the left, you can see the site with the different genetic varieties. On the right, you can see those are the clonal fragments. And then 8 months later, we went back, and I could not believe how well they were doing. They were growing over their base, attaching to the reef. They're branching, they're fusing, they're doing exactly what we want them to do, and even better. And so I actually am anticipating that some of them may be sexually matured next year, which will be 2 years from outplanting. So again, this is really good sign that our work is successful in terms of outplanting success after restoring these corals to the reef, that they're surviving and they're reaching, hopefully, these next critical developmental milestones. So as an example of how quickly we're able to upscale in terms of numbers of corals and diversity through assisted sexual reproduction efforts, In the last 2 years, in combination with Eric Bartel's teams for the staghorn coral species, we've outplanted 4 annual cohorts representing more than 20 unique families and more than 900 sexually produced genotypes, almost 3,000 corals, and across 10 different reef sites. And this is just one species. And so after outplanting, what I'll do is I'll conduct long-term monitoring to make sure, as I said before, that they're reaching these critical developmental milestones within expected timeframes, specifically referring to sexual maturity, so that we can document that these populations are becoming self-sustaining. So another successful example with a different species, with the elkhorn coral. So for this species, there are about only 500 remaining genotypes in the wild on Floater's coral reef, with less than 25 in the lower Floater Keys. So unfortunately, this species has been characterized as functionally extinct in this region, meaning the population is so small, it can no longer provide the ecosystem benefits and services that it's expected to, and it can no longer be self-sustaining through natural sexual cycles. But in the last 2 years alone, we've produced more than 400 new genotypes. So again, through these efforts, we're really able to upscale quite quickly. And then in combination with, alongside the restoration projects that I'm working on, I collaborate with different coral reef scientists in related fields, essentially for understanding coral fitness, stress tolerance, and seeing how we can propel the field of coral restoration science further even more. So as an example, I'm working with Erinn to understand the potential for trade-offs between important fitness-related traits like reproductive output and disease resistance. So fecundity is a proxy for reproductive output. It's like the number and size of eggs you reproduce. And we want to know, is there a cost to being disease resistant or susceptible on these other important fitness-related traits like fecundity? In another study, we outplanted sexual recruits of the staghorn species at a nearshore site near our lab, where this species historically existed, but in recent years has basically gone extinct at these sites. And even though multiple organizations over multiple years have tried restoring the site through the coral gardening idea, unfortunately, they eventually succumb to bleaching and die. So this year we ran a pilot test of outplanting sexual recruits that we knew harbored a special genetic variety of an algal endosymbiont that we know is more bleaching resistant. And so we outplanted them in the spring before the hot summer months where bleaching usually occurs. And what we found was quite astonishing. We found variation in response to thermal stress, where some of the genets bleached and died, but others actually bleached and recovered, and some didn't bleach at all. So right now, Dr. Courtney Klepak has taken samples across this time scale and is trying to understand the mechanisms underpinning the variation in the thermal stress tolerance that we see. But ultimately, this provides hope that through continued research, we should be able to restore this site in the future. Also, corals live in a close association with a number of eukaryotic and prokaryotic organisms that make up the coral microbiome. And Dr. Klinges has characterized the microbiome of different disease susceptible and resistant genotypes that I've actually used as parents in a special breeding design experiment. And then she'll go in and look at the microbiome of the offspring of those different crosses and characterize the microbiome across the different life stages of the coral, as well as in different environments that are relevant to restoration, like our field nursery or an outplant site. And finally, as I mentioned earlier, corals rely on a specific set of an abiotic environmental cues to regulate the timing of spawning and gamete release. But there's also some evidence to indicate that they use sexual cues, perhaps pheromones, to also regulate the timing of gamete release to make sure that they're releasing the sperm and eggs at the same time. And this is really important for species that are gonocores, meaning colonies are not hermaphrodites, they're not both male and female, but they're male or female. And usually the males will spawn before the females. But they need to be able to, regulate that timing so that way they're not wasting their gametes. And so right now we're looking into seeing if we can identify those cues, which could potentially be used as a tool for inducing corals to spawn more synchronously, which would effectively support better, more successful fertilization. And ultimately, all of this work and collaboration goes towards more effectively and more quickly restoring Florida's coral reef. So that way we can see a return of these essential ecosystem services and functions that not only do we humans rely on, but the natural world as well. And with that, I'd like to thank you for your attention, and as well as the mentors and the sponsors and collaborators and students that have supported this work.
Thank you so much, Hannah and Erinn. That was absolutely fantastic. Your work is amazing, and there's lots of really good questions, and we do have a little bit of time for questions. So I actually wanted to start with a question that came through in the chat. So everybody may not have seen it, but I think it's a good question. Erinn did answer it, but I think we'll start there. And the question comes from Heidi Ellard, and it's, does the coral keep growing at the same pace after the first year? Erinn, do you mind answering that for everybody who may not have seen it in the chat?
Sure. So we have been putting a lot of effort to document these growth rates in our nursery. So as we fragment the corals and then as they're being grown out, we have projects kind of quantifying species diversity and even genotypic diversity related to those growth rates and how long they last. And so we know the first year they seem to be quite accelerated compared to like if we had kept the corals intact. And then we also did a study that's published that quantifies the growth rates of the corals after they've been outplanted. And that was about like 1 year post outplant compared to of the microfragments compared with a larger kind of previously used size coral. So maybe like softball size. And we know that they grow 50 times faster within the first year after outplanting too. But we don't know what happens long-term. And we do think that there is a plateau because these corals are obviously moving resources away from things like growth potentially and into reproduction, because Hannah's work has shown that these corals are reproducing. So they're probably, plateauing a little bit in their growth rates in order to be able to still divert that energy into a gamete creation and spawning. But TBD on long-term stuff.
Right, so you're going to track some of the colonies over time? And
yeah, we're trying to, and we're integrating a lot of like 3D photogrammetry now into our outplant sites because it's really difficult to quantify the 3-dimensional growth structure of corals, especially once they get out into the wild and start branching. And so we actually have staff now that really focus on integrating the 3D modeling approach to get a better idea of their growth in situ.
That's great, thank you. A question from Neil Glickstein. Have you looked at predation to outplant individuals when they are small and vulnerable, parrotfish, etc.? Is predation significant to your plot?
You want to take that, Hannah? Or?
I mean, yeah, I can address it. So we do know that, yes, predation is an issue and there are site differences. We typically see fish predation to be more of an impact with early outplants. And so we'll do 1 month monitoring and see evidence of the fish predation, but inevitably they always recover and survive. And so when we go out and we see evidence of that, at first it might be a little bit off-putting, but when you go back and do the 3 month or 6 month or 1 year monitoring, they're able to recover. And then later on, as they get larger, we notice that snail predation tends to be more of an issue. But again, it can be species specific, it can be site specific. And those are things that we are working on to better understand and develop ways for mitigating those issues.
Great, thank you. A question from Tim O'Donnell. For your bleaching resilience experiments, are you simultaneously genotyping the symbionts along with the corals to see how that affects the results?
So yes, we characterize the symbiont community at the same time as understanding the genetics of the coral host. We have a really unique kind of setting, I guess, for our nurseries. So our in-water nurseries, the corals out there, especially the staghorn corals, they all seem to shuffle their symbionts to be one type. And then a very similar thing happens in our land-based nursery where they all shuffle to be one symbiont type, but they're different. So the ones on land are actually, we're cultivating inadvertently known to us for years, we've been encouraging the acquisition of a really heat tolerant symbiont. So we're utilizing that kind of novel accident to our advantage, kind of in the example of Hanna's project with Courtney Kleepak. But yes, we understand that both the host genetics and the symbiont, the algal symbiont, influence heat tolerance and many other things as well. And so we try to characterize as much as possible when we're doing some of those experiments.
Thank you. That's great. A question from Jim Bacon. He's asking, what is the scope of the reef restoration needed to reverse the American, if not global, deterioration? I think that you addressed this a little bit when you were speaking, Erinn, because you talked about your study that you're doing throughout the Keys and said it would be about 20 years to restore it back to 1970 levels. But maybe you can comment on the scope of that work in relation to other efforts around the world to restore corals.
So from what I know, that Mission Iconics Reef effort that we're helping to lead is the largest targeted coordinated restoration effort in the world. And that is only 7 reefs throughout the entire Florida Keys. So there's plenty of reef habitat in between that we are working on, outplanting to actively, but there's a ton of area to even cover locally. And so, right now, I think our goal is we have certain areas that we know our restoration corals do really well in. And so we are focusing our efforts there and also these iconic reefs. But as we continue to do site selection, we have to think about things like reef connectivity, providing outplanting locations that are sources for larvae to move to other reefs that may be sinks, so that we can have a larger impact spatially as we focus our efforts on a smaller footprint. But I mean, I know other regions around the world, especially Australia, is taking on a huge amount of restoration efforts too. And they, have $100 million from their government to conduct coral restoration and integrate heat tolerance. And they're doing some really amazing work too. But I would say they have kind of an advantage because they're not working at the degraded state that we are within Florida's coral reef. I mean, we're coming back. We are trying to come back from the brink of extinction, whereas the Great Barrier Reef still many places has really decent coral cover. And so I think the lessons learned, at least there, is, don't wait 50 years like we did to try to bring corals back. As soon as you start seeing them come back and not recovering, you can take some action to try to help things along maybe earlier.
I have a question that kind of relates to your coral arc, and wondered if you can cryopreserve either coral gametes or embryos, and if there's a way to bank those.
Sure, I can address that. So yes, the technology has been developed to cryopreserve coral sperm. And I do know that scientists are working on other types of tissue samples like tissue or eggs or small juveniles as well. And there has been successful demonstration of producing offspring with fresh eggs and cryopreserved sperm. And so we're actually a part of a consortium that has demonstrated this for the first time using the Crabera palmata species, where fresh eggs collected from mothers in Curacao were crossed with preserved sperm from colonies in Florida, Puerto Rico, and Curacao. To show that we can produce viable offspring and rear them. And we're actually conducting a follow-up study to look at the thermal tolerance of those different populations. So it is possible to preserve the sperm and use that as a way of gene banking as well. And that's something that we are also hoping to develop as part of our restoration framework moving forward.
That's great, thank you. Can you maybe comment on how hurricanes impact coral reefs? It seems astounding to me that they survive that. And do they fragment during hurricanes? What's the general impact of a hurricane on the reef?
So I can speak, anecdotally from how it's impacted my work. So when I started, as I had mentioned before, I, at the very basis of doing asexual reproduction work, you need to have sexually mature colonies, which depends on the size. And so right before I started, Hurricane Irma came through and really had an impact both on our nurseries and our field sites. Some of the nurseries disappeared, some of the large corals disappeared, and yes, the branching species were fragmented. Now, these branching species rely on physical forces to naturally fragment and grow their population. But what we're seeing is with the intensity and frequency of these catastrophic storms that they're just decimating, you know, and they're really having an impact. And so something that I saw was I was looking for sexually mature corals. I went out to one of Mote's oldest restored population where the fragments had fused and actually had grown to the size to be sexually mature. So they were of the size necessary to be reproductively viable, and they were otherwise healthy. And I went out year after year seeing if they had gametes, if they were producing gametes or spawning. And 3 years in a row, I didn't see anything. So I was then thinking, okay, well, is there a case of chemical pollution here and developmental toxicity? But ultimately, what the study showed is that it's most likely an indirect effect and long-term impacts of Irma on the over fragmentation of those populations and how they just shunted most of their energy into recovery and growth and survival. And so we lost a few years of those corals actually reproducing. So in that way, hurricanes definitely have a negative impact on the reproductive cycles of our species.
That's really interesting. Thank you very much. We only have a few minutes left here, so I just have one more question, and then I'll turn things over to Chris to wrap up. And so apologies to everybody who we didn't get to your questions tonight. There are a lot of great questions that have come in. Could you use eDNA technologies, environmental DNA technologies, to monitor the biodiversity impacts of your restored reefs?
So yes, we actually have some projects in place now that are with Dr. Eileen Maldonado, who Hannah mentioned, and she is going to be using eDNA to look for cryptic species or to capture the impacts of our restoration efforts on a molecular level, which may give us more diversity within our information. So we're pretty excited to start using that moving forward to help characterize like the impacts of our restoration at that level.
Great, thank you so much. Thank you so much to both Hanna and Erinn. Absolutely fantastic work, really interesting. I'm going to turn things back over to Chris to wrap up for the evening. Thanks very much to the audience for joining us.
Yeah, thank you, Erinn, Hanna, and Andrea, for sharing your time, your work, your stories, and those gorgeous photos with all of us. That was fascinating. I'm getting tons of great comments and lots of messages coming through. Everyone loved it. I think there were a lot more questions. We could have kept going much beyond the allotted hour. So really, really grateful. Hopefully we can see you two join us in Gloucester for our science forum when that returns in the spring of 2022. Thank you again to our sponsors, Ms. Elizabeth Moore, the 1911 Trust, and James and Gail Bacon, and to everyone out there for continuing to tune in and to support GMGI's mission. Please stay tuned and stay in touch. We have more amazing science innovators lined up in the coming months. I'm going to post the December speakers date and reveal the name in just a moment. So stay on for one more minute so you can save the date. Good night, everyone, and we'll see you in December. Thank you.